So in our capillaries, we're going to have to exchange lots of substances. This is not just respiratory gases that we're worried about here. We're also worried about things like nutrients, so sugars, fats, proteins, waste molecules. So this is kind of where CO2 comes in, but also where we have things like urea from protein metabolism and lots of other waste molecules, as well as hormones. So dealing with all of the various hormones that we have in our systems, whether it's estrogen, testosterone, or any of the other hormones that we talked about in the endocrine system. Okay. We have to have exchange between blood and tissues for these. That's the whole idea of actually using the cardiovascular system to move the stuff, is that we're sending it to every tissue so that we can send a signal or start a signal in one place and send it to the next. Okay, so we're not just going to be talking about gas exchange. Instead, we have exchange of lots of things. Now, because of that, we're going to need lots of ways of moving those substances. Diffusion is great for things that are small and nonpolar, like your CO2s, like your oxygens, even to a certain degree, some nutrients and some hormones and stuff. However, for larger molecules or things that can't move across the membrane, we are going to either use bulk flow or vesicular transport. So now we know that diffusion means that we have substances moving from high concentration to low concentration. We can utilize diffusion to move things across our blood vessel membranes as long as the solutes are small and if they are non-polar. Okay, so if they're small and non-polar, this means that they'll be able to move through the cells themselves. They'll be able to move simply through the cell membranes and not have to pass through a transport protein. Remember, if something is large or if something is polar, it won't be able to make it through that non-polar region in the center of our membranes. So they're not going to be able to move across. However, things like oxygen and things like CO2 can. We don't need specialized membrane proteins or anything like that for those. So that part's great. That makes our life a lot simpler when it comes to just simply gas exchange. Okay. Now, larger solutes, so things like some smaller proteins and stuff like that, will be able to pass through the tiny holes in between the cells, or what we call sinusoids. They'll be able to pass through some of those fenestrations or some of those gaps. However, they will have to be still fairly small, not some of our larger proteins like our colloid proteins, like we're going to talk about here in a second. Now, for larger things or big stuff that we want to move in big chunks or things that are polar, instead we're going to use what we call vesicular transport. Okay. Now, vesicular transport is where we're going to basically take in little tiny vesicles of the substance. Okay. So they're going to come up to the cell membranes and they're going to kind of get invaginated by a little vesicle and get pulled into the cell, at which point the vesicle moves across the cell, finds the other side, and then releases the substance on the other side of the cell via exocytosis. Okay. This is how we're going to move things, especially like fatty stuff. So like your fatty acids, some of our hormones, this is where we'd move kind of testosterone, cholesterol, things that have those big ring structures. This is actually how we would go about moving those. Then we've got bulk flow. Bulk flow is how we're going to move small substances or even larger substances that are dissolved into the fluid themselves, but especially smaller substances that are dissolved into the fluid. We're going to move them by moving fluid. Okay. So simply by increasing the amount of fluid that we move, we can increase the amount of dissolved substance that we're moving. All right. Now, this is going to depend on kind of two opposing forces. We have hydrostatic pressure, which is basically the amount of force that the water is exerting. So you can picture this as like, okay, you put a coffee filter in here and you put some coffee in the coffee filter and then you start to pour water through it. Well, if you pour water slowly, you have a fairly low amount of hydrostatic pressure. Water will come out slowly of the coffee. However, if you pour lots of coffee very, very quickly, lots of fluid will then move through. So lots of fluid is lots of hydrostatic pressure. A little bit of fluid is little bit of hydrostatic pressure. However, on the opposite side of this, we have what we call colloid pressure. We have special proteins in our blood, so things like albumin, which are there to basically make the blood more viscous, make the blood thicker, at which point it's trying to inhibit the amount of loss of fluids. Okay, that's because these albumins and stuff like that aren't going to leave the blood vessels. Now, one of the ways that we can move the fluid out is through this process of filtration. We have tiny holes, those fenestrations and things like that. So we have small holes in the blood vessels, and we can just simply put enough force with the substances through it, and the substances will move through along with moving fluid. However, this is going to happen on the arterial end, because remember, this is where we have more pressure. More pressure means more stuff will get passed through. However, this also means that the vein side of the capillary is going to be fairly low pressure, which means we can actually absorb fluid here. So the venous end of capillary beds is where we're actually going to be absorbing excess fluids in these surrounding tissues. So say you have a area that is swollen or something like that, it's been inflamed, so it has a bunch of excess fluid there. We can actually take up some of that excess fluid on the venous end.